Conclusion. Increased complexity and efficacy of adaptations take on new meaning in the context of major mechanisms of genomic change, including but not limited to gene duplication in multigene families, transposable elements, VDJ recombination, somatic hypermutation, and gene regulation mediated by cell-cell communication and antigen presentation. This is a very different paradigm than the concept of gradual accumulation of random point mutations in protein coding genes. In other words, paradigms such as the modern synthesis, which were developed in the absence of subsequent empirical discoveries, require a rethink. The underlying principles of natural selection remain the same, whether we are discussing point mutations or the synergy of major mechanisms of genomic change. But those major mechanisms have far more organizing power. As illustrated by the 2017 publication of the molecular living fossil of RAG1 and RAG2, namely proto-RAG, significant pieces of the puzzle of reconstructing past events continue to fall into place. These concrete steps are good to know because they represent the empirical facts that test the validity of evolutionary hypotheses. But in the press of urgent specialized research goals, we often fail to step back and reflect on the big picture of what we already know and is reflected in the Updated Evolutionary Synthesis, UES. One does not need to fit all the pieces of a jigsaw puzzle into place in order to see the whole picture. Consider a thought experiment with the following sequence of events. 1. A gene duplication event generates a primitive cell surface protein, call this protein 1, that fortuitously immobilizes a bacterial toxin or a viral particle and thereby decreases its pathogenic potential. 2. Additional gene duplication events and point mutations generate a small family of related proteins, subfunctionalization. Call these protein 1A, 1B, 1C, etc. Protein 1B fortuitously binds the toxin or virus more effectively, or bind more effectively to new variants of the virus. 3. Fortuitous insertion and movement of a transposon resembling proto-RAG results in a recombination event that joins protein 1B transposon to a nearby signal peptide of a secreted protein. Now, we have a secreted version of protein 1B that binds circulating toxin or virus more effectively. 4. These fortuitous events lay the groundwork for subsequent gene duplication and transposition events, and gradual refinement and fine-tuning of these mechanisms, and the primordial immune system continues to increase in complexity and efficacy by means of natural selection. For purposes of this discussion, the specific sequence of imaginary events is not important. They do not represent actual facts. Other creative listeners can undoubtedly bring their own knowledge to bear on the subject and come up with scenarios that are more plausible or consistent with known facts. Cause and effect become blurred in these scenarios. It is equally valid to say that during the course of evolution of the immune system, gene duplications laid the groundwork for VDJ recombination, or to say that VDJ recombination laid the groundwork for additional gene duplications, or to say that VDJ recombination laid the groundwork for primitive forms of somatic hypermutation, and so on. At each step in the process, we are witnessing the role played by natural selection in molecular evolution. Accumulation of useful variation is taking place. When combined with the more modern perspectives of Emergent Evolutionary Potential, EEP, shape-specific interactions and molecular binding events, SSM-IBE, and other modern evolutionary perspectives, the sheer organizing power of natural selection is clear. Major mechanisms of genomic change blur the distinction between gradual quantitative accumulation of changes and relatively sudden leaps in complexity, organizing power, and qualitative innovation. I would argue that the UES is well supported by both extensive empirical data and by a rational conceptual framework. Biological evolution by means of natural processes is more plausible and more fascinating than ever before. The metaphorical blind watchmaker continues to thrive. The effects of EEP on evolution can be seen at every level of complexity, including fascinating Darwinian examples of both coevolution and sexual selection. Consider, for example, the following excerpt from Sir David Attenborough's narrative of the beautiful film series titled Our Planet. Male orchid bees need a rich perfume with which to impress their females, and the orchids provide it. But the bucket is slippery, and the liquid into which the bee has fallen is slippery. The only way to get out is through a narrow tunnel. As it emerges, the bee is gripped tight, and that gives enough time for the plant to glue pollen sacs on the bee's back. As we approach what many have called the singularity, a perilous time when the very survival of our species can no longer be taken for granted, there are both spiritual and practical reasons why a deep understanding of the UES among the general public, including voters, is essential. Fortunately, the UES offers a more plausible explanation for evolution than ever before, thanks largely to an abundance of sound empirical evidence describing the inner workings of cells. Global warming is arguably the most urgent and important of the practical reasons for the general public to understand evolutionary principles. Interdependent species, including humans, simply cannot evolve fast enough to offset the life-threatening environmental consequences of global warming and other aspects of climate change. In the final chapter of this audiobook, I have listed several principles of the UES. This is, of course, not the final word on the subject, and I look forward to constructive feedback from the scientific community, as well as suggestions about how to better bridge the gap between professional researchers and theorists and the general public.